Hello, I'm Alfred Madere, and this is my team, and we are here to present about the current state of CRISPR technology and its applications both present and future. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR was first identified as a microbial immune system for its role in recording DNA sequences of viruses and detecting them using proteins like Cas9 and guide RNA. The discovery earned Emmanuel Charpentier and Jennifer Doudna a Nobel Prize. Scientists programmed the system to be able to cut and even insert new DNA at the matched location, giving it the ability to modify any gene. Here, the Cas9 enzyme in orange and the guide RNA in purple are working together to unzip and locate the desired DNA sequence via base matching. Once found, the system can either disable a gene by cutting and removing critical bases or insert a gene using homogeneous direct repair. The three-step process is shown. I'm Alfred, and I chose to focus on climate impact because I'm very interested in the methods of climate disaster prevention.
I'm Teddy, and I chose to focus on increasing livestock efficiency as food insecurity becomes a larger problem.
Hi, I'm Adrick, and I chose biofuel because it has to do with the environment, and it's a big concern, especially with climate change nowadays.
I'm Kyle, and I chose to focus on gene therapy because of its medical potential to save lives by treating diseases that don't have effective or safe treatments.
First, let's explore the extent to which CRISPR could combat the effects and causes of climate change via crop improvement. Current research shows that CRISPR has the ability to lengthen the shelf life of many produce products, increase the crop yield efficiency, and improve the climate resilience of several high-volume commercial crops. Apples brown because of the presence of PPO enzymes. By comparing DNA of apples that brown at different rates, scientists were able to develop and introduce an anti-PPO gene using CRISPR. These modified apples are called Arctic apples, and they are available in stores today. While Arctic apples were one of the first CRISPR-modified foods to reach stores, other companies are generalizing the process to other produce like potatoes and mushrooms. These recent success suggests non-browning gene editing could be generalized to most fruits and vegetables in the future. So why does engineering non-browning fruits matter? Food waste is a significant source of greenhouse gases, producing over 2.2 gigatons of CO2 annually. A significant fraction of this is due to expired shelf life, which could be dramatically reduced by CRISPR-modified products that are designed to last longer with anti-browning edits or other improvements. Scientists have successfully engineered wheat to be resistant to one of the most common crop viruses called Potosphaera xanthia that causes white mildew to develop and render entire harvests unfit for sale. The study was successfully repeated with tomatoes. Interestingly, the disease-resistant gene modifications also resulted in higher yield, leading researchers to believe increased yield modifications are possible across most plants. Now taking a look at the orange part of this graph, we can see that the unsold crops account for 55% of wasted food carbon emissions. When crops are unfit for sale due to disease or ruined by intense climate conditions, the energy cost of cultivation is wasted and greenhouse gases are released as they decompose. Much of this could be avoided with CRISPR-modified crops. A study was able to genetically modify tomatoes to have increased tomato response and high drought tolerance by modifying the OST2 gene. Significantly, similar process was successfully used on rice, an extremely climate-sensitive crop. Separately, scientists successfully created leaf-rolling genotypes in rice and observed superior drought-resistant characteristics. This is important because traditional rice will struggle to survive during impending extreme weather events. Higher survival rate of crops is necessary in an age of climate change because weather stress tolerance will need to be overcome in order to continue feeding the population, and energy efficiency will become more imperative, not allowing for wasted food producing greenhouse gas. Virtually every study involving CRISPR is in its proof of concept stage, so be wary of many extreme claims on the internet about what CRISPR can accomplish. These are my sources.
Lens looks at how CRISPR can be used to modify livestock to increase efficiency and animal well-being. CRISPR can increase farmed animal yields, improve the well-being of animals, and increase disease resistance in livestock. Food insecurity is a significant problem globally, as 1 in 9 people don't have access to sufficient food. And as populations grow, animal product production needs to raise by around 70% to meet these demands. This graph illustrates the growing food insecurity globally. One gene edited to increase yield is myostatin, which is a muscle growth inhibitor that can be disabled to increase lean meat yield in livestock. However, it can create mobility issues in larger animals due to muscle content. SRY is a protein that determines sex. If enabled in female cattle, they inhibit male traits, which allows them to grow faster and larger while using less feed. In a Japanese study, CRISPR was used to disable the myostatin gene in red sea bream, which resulted in a 16% increase in edible tissue. The American company, AquaBounty Technology, inputted a growth gene from Pacific Chinook salmon that caused the fish to grow larger in half the time of non-transgenic salmon. Dehorning is a process used in 93% of United States farms and is seen as an animal welfare concern, as it requires a cow's horns to be forcibly burned out of its head in order to protect animal handlers. However, by inputting the pulled genes from Angus cows, which don't naturally grow horns, geneticists were able to breed dairy cows that didn't grow horns. Porcine reproductive and respiratory syndrome is an endemic virus that costs the United States and European Union $2.5 billion a year. However, by silencing the macrophage protein CD163, infected pigs showed a lower viral load and lower mortality rate. African swine fever is a virus from sub-Saharan Africa that is spreading to Eastern Europe and has a mortality rate up to 100%. It has no traditional treatments, but geneticists have inputted the RELA gene from warthogs in an attempt to combat the virus. The process of bringing a transgenic animal to the consumer market is lengthy due to FDA regulations and long gestation and maturity times of subjects. Currently, only 2 genetically modified animals are cleared for sale, being AquaBounty salmon and an allergen-free pig. The length of time for studies and high failure rates also makes it difficult for studies to get funding. These are my sources.
Biofuel. So for biofuel, CRISPR can modify the genes of microorganisms to improve their metabolic rates, improve lipid content and biomass productivity for plants, which also increases metabolic rates, and use these boosts to create biofuel ultimately. So gene editing is mainly used for biofuel production, and this is through CRISPR's impact on biofuel and its ability to achieve a higher product yield for microorganisms with metabolic rates and plants which are contributors to biofuel production. Its gene editing capabilities will enhance these living organisms to help increase product quality and ultimately help increase biofuel production, along with staying efficient. These plants and organisms take in chemical sugars and nucleic acids, and their metabolic system, whether it's cellulose or lipids, whatever breaks down these components, it goes through that process. However, the Cas9 protein allows for the metabolic rates to be increased through gene editing, and it produces butanol, ethanol, and many other biofuels as the output. Some applications of CRISPR are for sure on microbes and plants. For microbes, it utilizes a Cas9 protein inside the gene editing process to regulate the gRNA being produced in order to increase higher product concentration and improve lipid content quality. These microbes can now produce more through the use of CRISPR. And for plants, it can modify the cellulose inside of the plants to improve product yield and produce high-quality biofuel ingredients, such as butanol and ethanol, through the energy breakdown process of the plant. So the most popular form of gene editing is through algae, and it utilizes algal biomass production to produce energy-dense compounds such as butanol that can replace fossil fuels. And it goes through the same process as described earlier. However, it's with algae, and it's one of the most popular forms since it's really easy to manipulate with algae. The transcription factors in the algal DNA, which limit lipid production, are also knocked down by CRISPR, so the metabolic rates will increase as well for higher biofuel production rates in algae. The current state and limitations for this is that biofuel can only meet 10% of the world's energy demand. However, CRISPR has proven to help with that and make biofuel production more efficient. Also, it requires more agriculture since biofuel is made through crops and utilizes crops. We're going to have to use more land in order to create biofuel. And these are my sources. Thank you.
This is my lens on gene therapy. CRISPR can delete, add, and replace genes to treat genetic diseases in humans. The forms of CRISPR are the Cas system, base editors, and prime editors. The Cas system makes a double-stranded break in DNA which can be not toxic, so there is a research into base editors and prime editors that only cut a single strand, avoiding the toxicity problem and making them safer. Different forms of modality have different advantages. Viral vectors, while being efficient, have higher immunogenicity than non-viral vectors, and the most common one, AAV, has a low carrying capacity for gene sequences. Non-viral vectors are minimally immunogenic, but are not as delivery efficient. CRISPR treatment of LCA to repair vision is in a clinical phase. CRISPR edited the photoreceptor gene to create functioning proteins. With a low dosage, some people regain partial sight without significant side effects, but it is far from complete. CRISPR is in clinical testing to treat lung cancer. T cells, which are essential in immune response, were edited to be able to target cancer cells. Edited T cells remained for the 9 months they tested and attacked cancer cells. Despite rare off-target effects, target area mutations were over 70% but decreased with time. The results of the few of many trials show the short-term safety of CRISPR. However, optimal effectiveness of the treatment is far away. Prenatal gene therapy overcomes some obstacles of normal gene therapy, such as avoiding an immune response due to an immature fetus immune system. The size of the fetus also means easier vector access to different target cell types and with more vectors. It also may treat severe congenital diseases before disease onset. In preclinical testing of Cas9 and base editor 3, a study shows the successful editing and sustained edited cells in the liver of mice at a rate of 40% after 3 months with no abnormalities or edited cells in other organs. CRISPR unintended and off-target effects pose a challenge to safety. Forms of modality can lower the rates of off-target effects from its over 50%, and these effects include toxicity leading to cell apoptosis and harmful gene mutations. Immunogenicity is basically the immune response rate. CRISPR is from bacteria that most humans already have antibody and T cell responses to, leading to potential decreased effectiveness and safety. CRISPR has shown potential to be an effective tool for treating certain diseases. However, with trial results being so new, it is hard to assess the long-term risks and results that come with the treatment. CRISPR and modalities of CRISPR are being worked on for safety and effectiveness in the clinical and preclinical phases. And here are my sources. This is our claim of fact. CRISPR can create more efficient and sustainable crop and livestock yields, increase shelf life, reduce food waste, and create biofuel, contributing to a more environmentally stable world. CRISPR can also start to treat genetic diseases in humans, but research limitations on genes of humans, plants, and animals, and safety concerns keep these applications out of public use for now. And we would like to open this to questions. And thank you for watching our presentation on CRISPR.